The objective of the following experiment is to observe the in vivo development of individual forebrain neural progenitor cells through time-lapse live imaging of zebrafish embryos. This is achieved by electroporation of DNA constructs coding for fluorescent markers that label sparsely positioned forebrain neural progenitor cells. Electroporated embryos are anesthetized, mounted in low melting point agarose, and imaged with time-lapse confocal microscopy. The results show the in vivo development of a particular forebrain neural progenitor cell.
This method can provide insight into the in vivo development of forebrain neural progenitor cells. It can also be applied to other regions of the central nervous system, such as the midbrain, hindbrain, and the spinal cord.
The main advantage of this method over existing methods such as single-cell stitched DNA or RNA injection is that this targets precisely to cells of interest at any given developmental time point.
This protocol begins the day before the planned electroporation. In the morning, collect 50 fertilized embryos from mated zebrafish. Bathe the embryos in embryonic medium containing phenylthiourea, or PTU. Incubate the submerged embryos for 22 hours at 28.5 degrees Celsius. The following day, at 22 hours post-fertilization, the embryos are ready to be electroporated. Begin by dechorionating the embryos manually with fine forceps. Transfer the dechorionated embryos into 10 milliliters of electroporation ringer solution containing PTU. Then, to anesthetize the embryos, add 420 microliters of tricaine stock at a concentration of 4 mg per milliliter to the solution. The embryos are now ready for electroporation.